Hello ladies and gentlemen, welcome back to C.A.R. Entertainment. I'm your host Alan Hamilton, and today guys we're going to be trying to translation, which is a process under the protein synthesis in A22 biology. So for further I'm just going to go straight into it. It's not a huge long PowerPoint, but it can be quite complicated to talk you through this. So we'll firstly go through by talking about the process, and then we'll go about the extension, and I'll talk you through that diagram which relates to the process. So step one of translation is that it carries on from transcription. So you have your mRNA that happens in transcription, and then it's taken to into the cytoplasm, and that's where the process starts in translation. So a ribosome will attach to the mRNA by its start codon, which is pretty much always AUG. That's the codon. And this is on this thing called the A site. So the ribosome comes to the start codon AUG on the mRNA that was synthesized in transcription, and this is called the A site where it attaches. A tRNA molecule with a complementary anticodon will then come into the ribosome with a complementary anticodon which will pair with the complementary codon AUG on the A site. The tRNA will also carry an amino acid to the ribosome. So as the ribosome will move along the mRNA to cover both these codons, the AUG and the anticodon, the tRNA with the complementary anticodon will pair up with the next codon on the mRNA. So essentially, it has AUG in there, the ribosome will cover it, anticodon comes in with tRNA, and then it will move along, and that tRNA with the complementary anticodon will pair up with the next codon. So after AUG, let's say it's AGC, let's say that's the next one. It will then, the next tRNA molecule with a complementary anticodon will pair up with that one. So again, the tRNA will carry the amino acids, so that will bind in. So at the moment you have AUG and AGC in two sites, and you have two tRNA molecules with complementary anticodons binding together. So now, whenever the second tRNA molecule comes in and binds to that AGC, for example, you then have your first your start codon AUG. It is now no longer an A site but the P site. But the other one, which is your AGC with its complementary anticodon, is then in the A site, which is free for the side amino acid. So a peptide bond will then link the first two amino acids that the tRNA has to form a dipeptide. A ribosome will then notch along another codon, setting the tRNA free. So the tRNA that was in that carried the second amino acid will now occupy the P site. So it is now in the site on the far left, essentially, leaving the A site free, which is to the right of it, where a third amino acid can be brought into place by a tRNA to bind together with the codon. So essentially, if you look at the diagram above you, you see where the free A's bind to the free U's. That right there, ladies and gentlemen, is the P site, and then your A site is the CCG pairing with the GGC. Okay, then you have another molecule coming in, the AGT, that is coming in, and that will come into the A site. So your AAA will be broken off, and then after the peptide bond's made, meaning CCG will become in the P site, and the AGT that's coming in will then go into your A site. So the extension essentially is that the process will continue until your polypeptide sequence is built up and until a start codon is reached, meaning that newly synthesized polypeptides are formed by translation, and which are then transported to the Golgi apparatus in vesicles where they will fuse with the convex face. Polypeptides will then be produced and produce final proteins before being pinched off the concave face, and they will be transported by vesicles for use within the cells, or they will be excreted. So there's a thing called the one gene, one polypeptide theory, and this is whenever a man called Crick had a central dogma theory, which is at the bottom there, and then we'll go through the left diagram first, where you have DNA, which is then free transcription made into RNA, which then free translation is made into proteins, but DNA replication always happens over and over, and that's where you get more DNA from, so it doesn't run out. And essentially that's the exact same thing that happens in the second diagram you see there, only there's a thing called reverse transcriptase, which can turn RNA into DNA and DNA copy, and you can also have RNA replication in there as well. But essentially what this one gene, one polypeptide theory means is that if you say one gene, one protein, this isn't actually quite good, because it's only appropriate whenever a protein is only one polypeptide, and this is less suitable because usually there's more than one polypeptide. So You then might say one gene, one enzyme, but this is just a simplification from the reason that we just said. And this doesn't work because not all proteins are enzymes. Proteins can also be chemical proteins in a membrane. So instead, we're supposed to use one gene, one polypeptide. This is the most accurate link between DNA and their products. DNA is a control of enzyme activity. Thanks for watching this video. I'm Dylan Hamilton from C.A.R. Entertainment, and I'll see you next time.